Hello everyone, so this chapter is the continuation of the nervous system and it's going to focus on the brain. So let's get started. This picture here is showing major divisions of the brain. So if you look here, this part that's called cerebrum, it looks like a helmet. This is the cerebrum, and then there is this part called cerebellum. Cerebellum is also known as the small brain. So you have the cerebrum and cerebellum, don't confuse these two. And then there are some other areas. There's something called diencephalon, midbrain, pons, and medulla oblongata. So if you were to take the cerebrum off and look at just this middle part, that's what's known as the brainstem, and it's made of medulla oblongata, pons, and mesocephalon. Mesocephalon is the midbrain. So mesocephalon, pons, and medulla oblongata. Medulla oblongata is going to connect the brainstem with spinal cord, and then the pons acts like a bridge. It's going to connect the cerebellum here. So it's going to connect the cerebellum with the brainstem. And then the midbrain, is also known as mesocephalon, is going to process visual and auditory information. And then we have another area. It's really deep inside the brain. That's called diencephalon. It's going to lie deep within the brain, and it's going to attach mesocephalon to cerebrum. So it's going to attach mesocephalon, which is this midbrain, with the cerebrum, which is this helmet-looking like part. And then diencephalon is going to be further divided into 3 subdivisions. We'll talk about it when we get there. This picture is showing the sagittal section here, and also the coronal section, which is also known as frontal section. So in the sagittal section, you can see the medulla oblongata, and this is going to connect with the spinal cord. There is this part that looks kind of like a leaf or tree root, that's known as cerebellum, and then pons acts like a bridge between the cerebellum and the brainstem, and then there's the mesocephalon known as the midbrain. And then on top of that, we have the hypothalamus. That's the control center of the brain. Above the hypothalamus is thalamus. Thalamus is what's going to relay sensory information. So if you remember from last chapter, interneuron would go up and connect to the thalamus, and there'd be another neuron that's gonna, a third level neuron that connected from the thalamus to the higher centers of the brain. So that's thalamus. And then there is this portion that's called epithalamus. This thalamus, epithalamus, and hypothalamus are part of the diencephalon. So diencephalon have 3 subdivisions, and those are these: thalamus, hypothalamus, and epithalamus. On this picture, you can see the cerebellum, medulla oblongata, pons, midbrain, and then there's some spaces. These spaces are known as ventricles. So we're going to talk about this in upcoming slides. Okay, so brain is made up of tissues that have 2 colors. That's going to be gray matter and white matter. So there's gray color and white color. When we talked about spinal cord, we also had the same tissues. We had gray matter there and white matter. Now so that's similar. They both have gray matter and white matter. Now what's different is that when we talked about the spinal cord, the gray matter was on the inside in the spinal cord, versus the brain where it's on the outside. So on the outside you have the gray matter, on the inside you have the white matter. And again, this gray matter is consisting of the cell bodies. That's where the cell bodies reside. And then white matter, that's where the myelinated axons are. And myelin is lighter in appearance, and because the reason for that is because it's made up of lipids. And if you remember from the general nervous system chapter, these myelin sheets were formed by oligodendrocytes in CNS and by Schwann cells in PNS. So this picture here is showing you the ventricles of the brain. So there are 4 ventricles total. Ventricles mean spaces. So there are 2 lateral ventricles to the side. So these are the lateral ventricles. And there is the 3rd ventricle and then the 4th ventricle. There's only one of the 3rd ventricle and then one of the 4th ventricle. Now these ventricles, if you look here, they have this hole here that's going to be known as interventricular foramen. And then these lateral ventricles go down, and then you see the 3rd ventricle is connecting to the 4th ventricle, and where it's connecting, that's called cerebral aqueduct. And in between these two lateral ventricles, there is this wall that's known as septum. Septum is a wall pellucidum. So you have septum pellucidum and then cerebroaqueduct here. So I said the space makes these ventricles. So the spaces are known as ventricles. And these ventricles are filled with fluid, and the fluid that is filled with is called cerebrospinal fluid. You'll see most doctors calling it CSF. And CSF, we talked about it in the last chapter too. It's found in brain and spinal cord. When they want to take some CSF to test for infections, they don't want to take it from the brain, even though it's found in the brain and the spinal cord. They take it from the spinal cord, and they take it from the spinal cord with a spinal tap from the area where there are only nerves and where the spinal cord ends. And the reason for that is because they don't want to damage the brain or the spinal cord. So looking at it again, you can see the two lateral ventricles in between. In between is the septum pellucidum. So septum pellucidum, that's going to be connecting these two. And then there's the third ventricle, aqueduct of midbrain, and then the fourth ventricle. So these are interconnected spaces that are filled with cerebrospinal fluid. Another thing we have that these are lined by epidermal cells. And if you remember from general nervous system, epidermal cells are type of neuroglia cells. So these are the supporting cells that are found in the brain. And their job is to secrete CSF. So those epidermal cells are going to line inside of the ventricles, and then they're going to produce CSF, and these ventricles are going to be filled with CSF. Third ventricle is surrounded by the walls of thalamus, so because if you cut the brain in the middle, you have the right and the left thalamus. And the fourth ventricle is between the pons and cerebellum, so between the pons and cerebellum here. Here's another picture showing you the ventricles. So the lateral ventricle, third ventricle, cerebroaqueduct, and the fourth ventricle, and there is the septum pellucidum between the lateral ventricles. So there are 4 of them. And the function of the CSF in brain is to cushion the brain and also move the chemicals around. So like I said, CSF is not only found inside the ventricles, but also outside the brain and in the spinal cord. So if you hit your head not too hard, CSF will cushion it a little bit. And this is what they look like. Now, how they found out this is what they look like, the first person who discovered the shape of these ventricles was Leonardo da Vinci. He was an artist, so what he did, he drilled a hole in the brain of a dead ox, and then he filled it with melted wax. So he injected melted wax in the spaces of the brain after drilling a hole, and after that he dissected all the brain tissue, and what was left was these shapes. So that's how he found the ventricles, the shape of the ventricles, without any science knowledge. This is really amazing. Okay, so what's covering the brain? So the first thing that you got under your hair is going to be the scalp. So the scalp is under your hair. And then you have the occipitofrontalis bone, and there is epicranium, which is connective tissue. So again, you have the scalp, and then you have your skull bones. So if you were to cut into the bone, the first thing you will see are these membranes, and that's what's going to cover the brain. And these membranes are called meninges, and there are 3 of them. If someone have meningitis, so meningitis, which is inflammation of the meninges, so when they say someone has swelling of the brain, technically it means that they have swelling of these membranes that are surrounding the brain. And then there's the cerebrospinal fluid that's going to cushion the brain. So meninges, there are 3 layers. There is a dura mater, and if you were to dissect sheep's brain, this is the first layer that you would take off. And this covering looks opaque fibrosis, and it's going to cover the outer part of the brain. And if you peel it or remove it very carefully, what you're going to see underneath is arachnoid mater. That's the middle layer. In between the wrinkles of the brain that you see, so those noodle type wrinkles, so that's where it's found. Mater means layer, and arachnoid refers to a spider, so it looks like a spider web. And then the pia is the innermost layer, and it's going to be directly on the surface of the brain. So if you look at this picture, if you take off the scalp, you use a bone saw, maybe take it off. If you cut into it, you're going to see this whitish membrane that's going to be called dura mater. So that's called dura mater. And then underneath it, you're going to see these dark lines, and that's called arachnoid mater. And arachnoid mater is where you're going to see surface blood vessels, and then the pia mater is going to be directly on the brain. Another thing I wanted to say, the appearance of the brain is fairly gelatinous. That the brain that's been preserved, it's going to be fairly hard. Actual brain is fairly gelatinous. So the way mummies were preserved in ancient Egypt, they took out all the organs out of the body to keep the body from decaying too quickly, and then they put the organs in the jars. And they would also take out the brain, and like I said, it's if it's been preserved, it's gonna be fairly hard, but the actual appearance of the brain is fairly gelatinous. So the way they took out the brain, if you remember from the bones, there's a cribriform plate. It's a really thin bone. So they would go into the nose, and there's this really thin sheet of the bone, and on the other side of the bone is the brain. So they would take a straw, put it through the nostrils, and suck the brain out, and then they would bury the organs with the pharaoh. Okay, so I wanted to talk a little bit about amoebic infections. So this is an organism that's called amoeba, and it's a protozoan. And the reason I want to talk about this is because there's always a recent case whenever they talk about amoebic infections. Again, it's found in pond water. It's a freshwater organism. And whenever there's a case, news people, they usually describe it as a brain-eating organism. So what it does, it's going to cause inflammation of the meninges. So it's going to cause meningitis. So if you go swimming in a place where there's water that's been infected with this organism, or where this organism is living, it's gonna go into your eyelid maybe, or the nose, or you might swallow it. It's gonna make its way into the spinal cord, and it's gonna cause meningitis. And that's again inflammation of the meninges. I don't expect you to know this life cycle. I just wanted to show you that it's gonna cause infection of the meninges. And then on this picture, what you can see is that they're doing a spinal tap. So the reason they're doing this, they do the spinal tap, is to get a sample of the CSF. Because there are multiple things that can cause meningitis, they won't really know for sure until they take a sample of CSF. And in this case, it can be bacteria, or it can be a virus, or it can be this organism that's called amoeba. So let's say if it's a viral meningitis and the doctors prescribe antibiotic, it's not really going to work because it's a viral meningitis. So this is the reason they have to take a spinal tap. And they're going to take this really long needle. They're going to insert the needle between L4 and L5. And once they insert the needle, if they hit the bone, they're going to back it up a little bit and then suck some CSF out. And then they're going to send the CSF to the lab. And in the lab, they can try to grow the organism and see if it's bacteria, or do some other testing to find out if it's a viral meningitis or if it's amoeba. In some countries, they don't do spinal taps. They say that it's really dangerous and it can damage the spinal cord. So if untrained people do the spinal tap, they can cause damage, but if a trained person does it, it's fine. So here's another picture of the bone, dura mater, this layer, and then you can see these dark lines. That's arachnoid mater, and you can also see these blood vessels there. Last portion of the lab, we talked about sinuses, and this is the superior top sagittal sinus that you can see here. Okay, so we talked a little bit about the functions of cerebrospinal fluid, and it's going to transport nutrients, chemical messengers, and waste products, and also it's going to cushion the brain. If you look on this picture, it's going to circulate around the brain and then also inside the spinal cord. And then it says that's formed by choroid plexus in ventricles. So what we learned that in the ventricles you have these epidermal cells, and those are the special type of cells that are going to make choroid plexus. And in this case, plexus is not referring to the group of nerves. So from last chapter, we were calling it brachial plexus because it was a group of nerves. So right now we're calling it the choroid plexus because it's a group of cells, epidermal cells. It's not a group of nerves. So it's not referring to the same thing. Now, if you go into the brain, there is a blood-brain barrier, also known as BBB. And BBB is made by these specialized helper cells that are called astrocytes. So these astrocytes are going to wrap around the capillaries and they're going to help maintain this barrier. So there are these endothelial cells that are held together by these junctions. We learned about tight junctions in the beginning chapters. They're going to hold these cells together. And then there are also astrocytes that are helping, kind of wrapping around the capillaries that are maintaining the barrier. So that's forming the blood-brain barrier. And this blood-brain barrier is going to prevent some chemicals getting into the brain, so the brain can only accept certain type of things. Some of us are a little more sensitive, and that the reason for that is because this blood-brain barrier varies from person to person. It prevents lipids and proteins getting into the brain. It allows glucose to get into the brain. And this is going to be inside the ventricles. So ventricles are lined by epidermal cells. So in the ventricles, there's these cells, specialized cells, that are going to form the choroid plexus, and that's what's going to secrete CSF. These epidermal cells that are held together by tight junctions are making the choroid plexus. These astrocytes that are holding the endothelial cells in place are making the blood-brain barrier. These epidermal cells are going to secrete the cerebrospinal fluid. So what this picture is showing you the structure and function of choroid plexus. So the epidermal cells are the barrier, selective barrier, that are transporting the nutrients. So on this picture, you can kind of see overview of the choroid plexus and the blood-brain barrier. There's the blood-brain barrier formed by endothelial cells that are going to be held together by tight junctions and astrocyte cells that are holding it in place. Then there are the epidermal cells secreting the cerebrospinal fluid, also held together by tight junctions. And on here you can see the activity of the ions and compounds. And then, so these are a group of cells that are together, and that's known as plexus, and this is called choroid plexus. So now let's talk about the parts of the brain itself, and then we'll talk about the functions. So the big part of the brain is, that looks like a helmet, I said in the beginning, is called cerebrum. So that's the cerebrum, that's the largest region of the brain. So us as Homo sapiens have the largest brain capacity, and the reason for that is because we have this area called cerebrum that's the largest. None of the other vertebrates have this. We have the largest cerebrum, therefore we are more advanced than the other vertebrates. It has two hemispheres, and these two are separated in the middle by this fissure. That's called longitudinal fissure. Fissure is a really deep groove. And then there is another fissure that's called transverse cerebral fissure, or just transverse fissure. And you can see I'm doing brain dissection. And this transverse fissure is going to separate the cerebellum from the cerebrum. So this part from this part. Okay, so the surface of the cerebrum has lobes. And these lobes are going to have the same name as the skull bones. And if you remember the skull bones, then you already know the names of these lobes. So there is the frontal lobe, and that's where you have the frontal bone, parietal lobe, occipital lobe, and then the temporal lobe. That's where your ears are. So we just named all four lobes. And then there's something called gyrus. Gyrus are these elevator ridges. So these ridges are called gyrus. So if I was just talking about this one, this is called gyrus. Now if we're talking about multiple of those, so plural, it's going to be called gyri. Then we have something called sulcus. These are going to be the depressions between gyri. So these folds, there is these depressions. So there's this depression right here, another one right here, another one right here. So those are called sulci. If we're just talking about one, it's called sulcus. So singular is sulcus, plural is sulci or sulci. So these long parts of the brain, they don't pull apart like your small intestine. They're held together by fibers, and those are called commissure fibers. So if you remember from last chapter, I was talking about commissures. That's where the information from the right half is going to the left half. So the left and right side of the brain is connected by commissure fibers. So the cerebrum is for higher function. Now let's talk about functions of the frontal lobe. Frontal lobe is the area where abstract thought is occurring, problem solving, thinking about the future, things like that take place. Things that other vertebrates are not capable of, for example, thinking about the future. And this is also the area where the primary motor cortex is, so that's where the conscious control of skeletal muscle is happening. So you can control when you move your arm or when you move your hand. That's happening in the frontal lobe in the area called primary motor cortex. If you were to look at this picture, we already labeled the longitudinal fissure. And then there are the gyri. We call these elevator ridges gyri or gyrus if it's a single one. Now there is something called precentral gyrus, and then there is the postcentral gyrus. So there's another groove that's going down through the center. That's called central fissure. And then the ridges that are in front of that are called the precentral gyrus, and the ridges that are after central fissure are called postcentral gyrus. And there's one more thing that's called lateral fissure. So there was the longitudinal fissure, central fissure, and then the lateral fissure. So the parietal lobe, it's going to deal with the information that's coming from senses. Not all the senses, but many senses go there. So it's conscious perception of certain senses, so not all of them. So the senses go there, and then from there they get analyzed, and your brain decides what to do with them. For example, temperature or pressure receptors to deliver information there. And then it's going to decide how to deal with that information, if any action needs to be sent out. And then we have the temporal lobe. This is where your ears are again. And not surprising, but this is going to deal with the information that's coming from your hearing. So that's called audition. So this is the conscious perception of auditory information. And then another information it's dealing with is the olfactory or the smell information. So once you pick up information, it's going to stimulate hair cells in your ear, and we will talk about it when we get to special senses. And then it's going to go to the temporal lobe, and it's going to decide what to do with that information. And then you have the olfactory nerve. That's also going to take the information for smell. That's going to be dealt with in the temporal lobe. Occipital lobe. Now this might be surprising for some people, but it's going to be responsible for the eyesight. So perception of eyesight or the visual stimuli. And if you look here, there's a large area that is devoted to eyesight. And we have really good color vision versus the animals who maybe live in the dark. They don't rely on their eyesight as much. They rely on their hearing. So the hearing area is more developed in them versus us. We have the occipital area that's more developed versus some other animals. Okay, so what we're going to talk about next is a system. So neurologists, they take little parts of the brain, they link them together, and they call it a system. So for example, hypothalamus may work with pineal gland for some of the things, and they're going to call it a system. So I'm going to give you one system that we're going to focus on that's called limbic system. And this is going to be responsible for emotions and forming memories, or for memory formation. Now, just for the comparison and contrast, I have put another system here, reticular formation. We're not going to focus on it as much. So the reticular formation system is going to be responsible for conscious actions and some of the actions that you don't really think about. For example, peristalsis or release of bile or your heartbeat. You don't constantly have to think about that. And the way they know this part of the brain is responsible for this thing, they can take an animal. So they can either use a case study, maybe like happened to somebody where they got damaged certain part of their brain, and then that created some type of difference in their behavior. So they can study that, or they can take animals and they can damage certain part of their brain, and then they can study the animals to figure out what is this part of the brain responsible for. So limbic system is going to include the structures in the middle, and that's what medial aspect means. So structures on the medial aspects of the cerebral hemispheres, and dicephalon. And in the beginning I said dicephalon has 3 subdivisions, so it's coming up on the upcoming slides. So first I'm going to show you the location, then I'm going to tell you what they do. So just to give you an overview, so we have the cerebral cortex here, and there is something called corpus callosum, and then there is the thalamus, and then the brain stem, and this part known as small brain is the cerebellum. So again, don't confuse cerebrum with the cerebellum. Then deep inside the brain, there's dicephalon. That's going to include thalamus, epithalamus, and then the hypothalamus. Here's another picture that's showing you the sagittal section through the cerebrum. This is also showing you the areas that are associated with the limbic system. And then on this picture, you can also see the hypothalamus. Above it, you have the thalamus. And then something that looks like C shape, and that's called cingulate gyrus. Okay, so diencephalon is going to include 3 parts. So let's just label that. That's what's going to make the diencephalon. Diencephalon lies deep within the brain, and 3 parts that are going to make the diencephalon is thalamus, pineal gland or epithalamus, and then hypothalamus. So for the limbic system, located in this area, there's this C-shaped structure, and that's called cingulate gyrus. It's going to look like a C. The cingulate gyrus is an anatomical feature that's going to help with the emotions and memory formation. So the limbic system, like I said, the neurobiologists, they take parts of the brain that work together and they make it a system. So the limbic system includes part of the diencephalon, not the whole thing, and this box shows you here the parts of the brain that are included in the limbic system. And now it can differ depending on who you're asking and how much research there's been in the area. This area is really still kind of new. They're still doing a lot of research, and depending on who you ask, there can be different interpretation. So it's going to include parts of diencephalon and some cerebral structures that are going to encircle the brainstem, and that's the brainstem there. So we have thalamus, hippocampus, amygdala, and hypothalamus. So the next structure that we have is the thalamus. This is the part of the brain that you can see here, and it looks like a bird egg, and there's a right and left thalamus. So it's paired. And the purpose of the thalamus is to relay and process sensory information. It relays and processes sensory information to the cerebral cortex. So it's like a distribution center. And for an example, Amazon has a distribution center. So you go online, you make your order, Amazon grabs that order, and it goes to the distribution center. Distribution center is going to organize everything, and it's going to send the package to the right address or to your address. So this thalamus, it works like a distribution center. It's gonna receive information, and it's gonna relay the information to the higher centers. So for example, information coming from the outside world about the weather, if it's hot or it's cold, what is the temperature. It's going to organize the information, kind of sort it out, and it's going to send it out. It's going to send it out to wherever it needs to go, maybe to the frontal lobe or the parietal lobe, wherever it's going to need to go. And the thalamus is going to decide that, and it's going to send that information there. It's also going to regulate sleep and consciousness. And then from last chapter, there was an interneuron, second level neuron that was bringing the information into thalamus, and there was a third level neuron that was going from thalamus and taking the information to the cerebral cortex. So to the higher centers of the brain where consciousness is. So how do we know that thalamus is responsible for that? So one would be case studies, or they can create a damage in animals' brain and see what are the behavior changes are in the animals. So this example here is a case study. Patient had damage to his brain due to stroke. A stroke is when the blood supply to the part of the brain is cut off. And if it doesn't have blood supply, that means there's no oxygen, and the brain tissue is going to die. So in this case, you have damage to the thalamus. So once you got dead brain tissue, in this case in the thalamus, you cannot process sensory information anymore. And on this picture, in patient 1, you can see that the right side of the thalamus was damaged. So this patient can have pain or paralysis in certain parts of their body. And since the right side of the thalamus was damaged, if you touch this person in these areas, instead of feeling finger or hand in these areas, he's going to feel pain. And that's because the thalamus is damaged and the body or the brain doesn't know what to do with that information. And instead of pain, this person can also have paralysis. So in patient 2, the right side of the thalamus is processed, and in this case there's paralysis in these 2 areas. And then if there is damage to the left side of the thalamus, maybe they lost sensation in their finger. So there's different things that can happen if you damage the thalamus, because the body doesn't know what to do with the information that's coming in, and there's no relay center that can distribute the information to the right areas of the brain. So this person is going to have difficulty processing sensory information, and that can result, the damage can result in partial paralysis or extreme pain. Underneath the thalamus is hypothalamus. It makes hormones. So there is the thalamus, underneath it is the hypothalamus. Hypothalamus is going to make hormones. We did not cover endocrine system, but in physiology you'll cover endocrine system, and you'll learn a lot about the hormones that are being secreted from hypothalamus. So these hormones go in your body, and they're going to cause other organs to secrete other hormones. For example, it can go to the adrenal gland. It can cause the adrenal gland to secrete another hormone in your body. It's the control center. And in this case, we can focus on the sympathetic nervous system, or fight or flight mode. And this is what's going to control fight or flight mode. If you're afraid or angry, this is tied into how you create emotions, and it enhances memories. So it's going to, hypothalamus helps maintain homeostasis through endocrine system. And the example you can think about is a mouse that's in a maze. Let's say you put a mouse in the maze, and it's going to take them a while to find their way out of the maze, maybe let's say an hour or so. And the reason for that is because there's nothing at stake. There's no immediate danger. They're calm. Their memory formation is pretty slow. Now, let's say a behavior biologist creates a maze with water. I found this example. So the rat has to swim randomly until it finds the platform, and it can't see the platform. So once it finds it, it survives. Now in this case, because the rat's going to die unless it finds the platform, so there's something at stake. So the sympathetic nervous system is going to kick in. It's going to allow it to remember and figure out the problem much quicker, because if it doesn't find it, it's going to die. So if there's nothing at stake, your sympathetic nervous system or fight or flight mode is not active. It might take you longer. If sympathetic nervous system is active, it's going to help to figure out the problem quicker. So think about maybe, when maybe someone, was in an accident, or if you were in a fight, you can remember those things very clearly, maybe even the clothes that you were wearing. Or if you try to remember something very emotional, you can remember every little detail of the events because your sympathetic nervous system was active and it... Okay, so hippocampus. There's another structure called hippocampus. There's this structure that looks like a pea called amygdala. And this amygdala is responsible for really extreme emotions. So for example, anger or fear. So really, really extreme emotions. You can design an experiment in the lab, where in these days they can use technology, take a little wire and stimulate amygdala in animals. When you do that, it's going to result in extreme emotions. So emotions that are not in a normal level, they're extreme. So really, really angry or violent, or behavior, or maybe a lot of anxiety. And that's how we know that amygdala is responsible for these fearful or angry expressions or emotions. Another thing they can do, instead of stimulating amygdala, they can completely damage it and see how it's going to change the behavior of the animal. So here, stimulation can result in increased anger, violence, and fear and anxiety. And then you can see if the same thing is happening, or maybe it results in something different. So you can study the behavior and how it changes after you damage something in the brain. So here's an example of it. So you can see the same thing happening, or it can be a different behavior or opposite behavior after you maybe create lesioning or damage the certain part of amygdala. So Kluver-Bucy syndrome is when people are born with it. So people are born with it, and some animals are born with it, but sometimes you can damage amygdala and it can result in Kluver-Bucy syndrome. Let's say you have a cat and you put a wire in there and you damage the amygdala. You let the cat heal, and then you observe the behavior. Now when you do that, you want to have a list of behaviors that it was showing before you damaged the amygdala, so you have something to compare to. So what you will see is there's a heightened sex drive, There's hyperphagia, which is eating. Hyper means more, phagia means to eat. So if you remember macrophages, to eat, big eater. So hyperphagia is increased eating. So it's eating more. Hyperorality is putting things in its mouth. And then also lesions in this area also cause you to have no fearfulness. So this in this picture, there's a monkey with the syndrome and it has lost its fear of snakes. So there's a snake over here. The monkey's trying to touch it. It's not afraid of it whatsoever. And that's because there's damage to amygdala. So docility, that means you're docile, you're passive. So you're not going to be afraid of anything. And then also there's reduced parental care. For us to pass on our genes, there's this behavior of caring about our next generation because we want to pass on our genes. And if there's reduced parental care, it's not going to help with that, with passing on your genes. So experiment with amygdala affects the emotions, but you can't say before you do the experiment you get the results what emotions are going to be affected. Amygdala is attached to this thing called hippocampus. It's called hippocampus because the name for the seahorse is hippocampus. Every seahorse species, the genus name is hippocampus. Hippocampus means seahorse. If you dissect it out, it's going to curl up like that and it's going to look like seahorse. So that's where the name comes from. So we talked about the parts of the brain that are responsible for emotions. So now we're going to talk about the parts of the brain that are responsible for memory formation. So in this case, hippocampus will take your short-term memory. In this case, that's what's occurring right now when you're listening to the lecture. So if I ask you what you ate for breakfast, you'd be able to tell me, or if you what you were wearing yesterday, you'd be able to tell me. But if I ask you what you ate for breakfast maybe 2 months ago, you wouldn't be able to tell me. That's because your short-term memory does not always go into your long-term memory. So that's the function of the hippocampus. So you can take something from today and you can try to stick into your long-term memory. You can take a shortcut where something traumatic happens, then you're going to remember that day even after a very long time. So if someone comes and punches you in the face today, you're not gonna forget that. So that's what I meant when I said if something traumatic happens, it's gonna create long-term memory really quickly. But then there are things like studying. So if you're trying to study for an exam the night before, it's only going to be in your short-term memory. It'll get you through the exam. But if I were to come back and give you the same exam maybe 2 months later, it's not in your long-term memory. You're not going to be able to perform the same way that you did when you took the exam day after you tried to memorize. So with that, you have to do a lot of repetition, and that's when it gets in your long-term memory. So what if you damage this? You damage this, then you can't get stuff into your memory. We call that amnesia. So if you forget who you are, that means all the memories that you made in the past are gone. You don't know what your name is. You don't know who your family is. You don't know where you live. That's called the retrograde amnesia. Retro is past. And then in really rare cases, some people can't make future memories. And that's what a blackout is if you drink too much. So that's when people drink like crazy. And then they get up next morning and then they can't remember anything about last night. And that's kind of a memory loss, and that's called anterograde amnesia. An example of this anterograde can be Dory from Finding Nemo. She can remember her name. She can remember where she lives, but forming new memories is kind of a task for her. And then there's a movie that came out a while back, and that's called 50 First Dates. So this is a really great example of damage to hippocampus. So Drew Barrymore in this movie got in a car accident, and after the car accident, she was unable to form new memories for a very long time. It's called 50 First Dates because every time she woke up next morning, she couldn't remember what she did the day before. So every day kind of reset every single day when she woke up. So she couldn't remember anything from the day before. So this has to do with the damage to hippocampus called anterograde amnesia. And then another example I have is this person called Mary Lou Henner. She's famous for her photographic memory. There are some documentaries on her that you can watch. She has photographic memory, so if you ask her what you were doing maybe in 1997 on this day at this time, she'd be able to tell you, which is really interesting. Or maybe if you ask her in 1995 what day of the week was January 1st, she'd be able to tell you because she has photographic memory. And when you watch her interviews, because there are a lot of shows that So there's some shows that she's been on and they interviewed her, and you might notice that she says that sometimes she forgets, and then you might think, oh, she has this photographic memory, how does she forget? When she says she forgets, what she's referring to, let's say if she has a script for a television show with many different edits, she doesn't remember which one to do, she remembers all of them. So most of us don't have that ability, but she does, which is really amazing. So earlier I gave you a slide where I drew a box around hypothalamus, thalamus, amygdala, and told you that this is part of the limbic system. There's also olfactory bulb, and so some of the neurobiologists, they might argue with you that, oh, this is also part of the limbic system. Olfactory bulb is cranial nerve number 1. That's right there. It's right about above your nose, your cribriform plate. If you smell something, that information doesn't need to travel very far to get to your brain. And the reason I'm going to go ahead and include the olfactory bulb into the limbic system is because sometimes emotions can come along with the memories. So maybe try to think about a very pleasant time in your childhood and try to think about a smell. Maybe you were around your grandfather or your uncle and maybe you associate them with the cigar smoke smell, or maybe you were in the kitchen and maybe your grandmother was cooking really, really good food. So that smell is associated with the emotion. When you smell that food again, you will remember those memories. So maybe randomly you're walking around and you walk past somebody who was smoking cigar, and the smell of the cigar is going to bring back memories from your childhood. So the smell is tied in with the emotions and also the memories as well, because it can trigger memories. That's because it's so close to the area where the limbic system is. And you can try to manipulate that. You can put out different type of smells if you're trying to remember something. So there's a question in your homework. It's going to ask you, how can you use smell in order to manipulate your ability to remember things? And this picture here is showing you the olfactory nerve. So that's the cranial nerve number 1, Roman numeral 1. Well, you're going to notice that there are these holes that are in the cribriform plate, and that's how the sense of smell is being delivered to the brain. A person who maybe gets punched in the face like a lot, and when they're smelling something, they smell a lot because they damage these little areas. This little bone here, it's very sensitive, and if someone's getting punched over and over, it's gonna create some damage. They can get these damaged, and these receptors are very sensitive here. Their sense of smell is shot because you damage these holes here that are taking these receptors, the information from these receptors to the brain. So if these are damaged, they're not gonna lose their sense of smell. So that's the limbic system. It's the main system that's involved with the emotions and memory formation. So once you have these emotions and memories, your brain has to decide what to do with them. So in this area, you have the interneurons that are taking the information to the frontal lobe. Prefrontal lobe is going to receive that information and it's going to interpret the information. Okay, so I mentioned the midbrain or mesencephalon in the beginning. So mesencephalon means middle. Cephalon is referring to the head or brain, so it's middle of the brain. It's below the diencephalon. So if you were to do a cross section, you're gonna see a little part that's gonna be looking pinkish, not exactly red, but pinkish, and that's called red nucleus. So the red nucleus is going to integrate information from the cerebrum, so this part right here, cerebrum, and cerebellum, that's right here, to maintain muscle tone and limb position. It's going to integrate information from cerebrum and cerebellum, and it's going to maintain muscle tone and limb position. So if you just sat there, you don't have to consciously think about falling over. Your body kind of takes over that and it controls that. So it's part of the midbrain, and that's going to do that autonomic function in the background. It's also going to contribute to the reflexes. So if you do something over and over, so we learned that reflexes can be genetic or it can be learned or acquired throughout life. So if you do something over and over, let's say you play piano over and over, then you kind of start remembering the notes. So you kind of play the first note, then you go to the second note, and then to the third. So you keep going. That's called muscle memory, and this midbrain is involved in muscle memory. So inferior to the midbrain is a bulge, and that's called pons. So pons is a bulge that's inferior to the midbrain. And this pons is attachment point for many of the cranial nerves. So you have to know these cranial nerves for your lab. There are 12 cranial nerves that you have to know. So there's the name of the cranial nerve, and then you're going to see there's a Roman numeral. So you're going to have to know both for full credit. So you have to know the name and the Roman numeral of the cranial nerves. Many of the cranial nerves are going to be located on pons. It's going to contain sensory and motor nuclei for these cranial nerves. On this picture, you can see this is the pons, and below it is the medulla oblongata. So we're going to talk about medulla oblongata on an upcoming slide. And then on this side, it's cerebellum. So it's attaching the cerebellum to the brainstem. So let's look at cerebellum first, and then we'll look at medulla oblongata. So cerebellum is this leaf-looking structure. It's below the occipital lobe. Sometimes it's called the little brain because there are a lot of functions that it's involved in. Sometimes when you're going back from work, you don't really have to think about constantly about the directions. You kind of just, sit in the car and you drive to your home. You don't really have to think about it a lot, and that's because this is coordinating the information, sending the information automatically. This is a bigger picture of the cerebellum. On here you can see there is white matter and gray matter. There's a lot more gray matter in the cerebellum versus the white matter. Again, white matter is where the myelinated axons are, and gray matter is where the cell bodies reside. These two parts, your lab is asking you to know the superior colliculus and inferior colliculus. And it's going to be located right where the pineal gland is, so right below the pineal gland. This is the cerebral aqueduct, and then there is the fourth ventricle, pons, medulla oblongata, and then the cranial nerve. So oculomotor is there. So this white matter tracks connect cerebellum to other parts of the nervous system, so the midbrain, diencephalon, cerebrum, pons, medulla oblongata, and the spinal cord. So this is the last part we're going to go over. That's called medulla oblongata. Medulla oblongata is going to connect spinal cord to brain. And the brain that's been taken out, sometimes it can get harder to decide where this medulla oblongata starts and where spinal cord starts. So this is passing through the foramen magnum on the occipital bone. So that's a big hole that you learned when you were learning bones. So below the foramen magnum is going to be the spinal cord, and above the foramen magnum is going to be the medulla oblongata. This is going to relay information to thalamus, and that's the distribution center. It's going to organize the information, send the information wherever it needs to go. So those are the functions of medulla oblongata. It relays the information to thalamus, and then thalamus sends the information to higher centers, and it's going to take the information from the spinal cord, midbrain, and the cerebral cortex to cerebellum, and it's going to regulate physiological functions such as heart rate, force of cardiac contraction, blood flow, and the respiratory rate. So some actions that are autonomic. So here you can see the pons, and below it is the medulla oblongata. So that's the medulla oblongata. And I mentioned reticular formation center, and that's also located there. This picture is showing the cranial nerves. These are part of the peripheral nervous system, and it's going to connect to the brain rather than the spinal cord. So there are 12 cranial nerves that you guys have to know for the lab. So you will have to know the names, and you'll have to know the Roman numerals for these. So these cranial nerves, they can be sensory only or motor only, or it can be both mixed. These are going to enter and exit the skull through different foramina. And I'm not going to go in detail of each nerve here because we will be covering these in lab. This picture is showing you where each nerve is going and the name. So vagus number 10. It's controlling a lot of organs versus some of these other nerves. This is a good picture. When you're trying to label, you can use this picture from your book, and it's labeling all the nerves. So the cranial nerve number 1 is olfactory, number 2 is optic nerve. So let's look at the optic nerve real quick. So optic nerve is bringing information from the eyes and sending it to the occipital lobe, but the information crosses over. So when we talk about special senses, we're going to talk about how the information is coming from the eye and it's being crossing over, and it's going to the occipital lobe of the brain. This is the acronym that you can use to memorize these 12 cranial nerves. Again, you have to know the name of the cranial nerve, and you have to know the Roman numeral of the cranial nerve in order to get full credit. So that's it for the brain chapter. Next chapter is going to cover the autonomic nervous system.